All right. Today I'm going to continue our discussion of leukocyte trafficking. I posted a new set of slides for today, but really they are the ones I didn't get to on Wednesday. So it's that same set. And this process, as I said last time, is this process of trafficking that can occur for a neutrophil or really any other cell type that would like to leave a blood vessel to go to a site of inflammation. This can be an activated, so an effector, or a memory T cell that wants to leave a blood cell or blood vessel in order to go to a site of infection, or this can be a naive T cell that wants to go to a lymph node and is going to leave a blood vessel. So everything I'm talking about right now is very much the general process for cells that are going to leave the blood vessel at a specific location. We made a parallel with this process to an idea of the highway last time. And we talked about how very quickly our cells are moving through the blood vessel, and the fact that if the cell just stopped in the blood vessel, its neighboring cells and surrounding fluid and surrounding proteins and such would collide with it at such force that it would basically shear the cell in half. So our cell is not going to be able to just stop mid-vessel, and is instead going to need to actually go through a specific process that has the stages that are listed here. The first stage of this process is the stage that I showed you in a little movie last time. That is a stage that is known as rolling. I was originally taught rolling as sticking. I like rolling a little bit better, but sticking also can kind of make sense. And so when I am driving on my highway tonight, and I get to my exit, first question, how am I going to know that I am at my exit? How am I going to know that this plot of trees in upstate New York is the place where I would like to leave the highway, as opposed to any of the other plots of trees in upstate New York that I will be driving by, because there's going to be a lot of them. What? There's a sign. So first of all, we need to have some way of marking this location in the blood vessel to indicate that this is a location where our cell wants to leave. So our different locations have to have some kind of mark or sign on them to allow for our cells to leave. So that's one thing we need to be paying close attention to. What else am I going to do when I get to my exit? There are kind of two steps that happen here at first. Yeah, Molly. Slow down. So I'm going to slow down, and I'm going to move over out of the center of the flow. And in fact, that's exactly what we see with our rolling cells in this process. And so I'll just quickly show you a part of that video again. This one. So our cells are going to be moving Come on, where's the one with the microscopy? So our cells, in general, these are our cells that are moving really fast through the vessel. They're moving so fast that you can't really distinguish them quite well. And we have these other cells that you can see have come to the side of the vessel and are moving slowly and rolling. They're moving slowly enough so that you're actually able to see them here. So they have pulled over to the side, got into the side lane, as well as slowed down. But what you will also see is that they are still moving. Of course, this is happening through protein-protein interactions. This is happening through some kind of biochemical interaction. And so what this will tell you is that the affinity of the proteins that are doing this interaction, or the molecules that are doing this interaction, must not be crazy strong, because it's still allowing the cell to move. If this was a crazy strong interaction, then our cell would get get stuck and wouldn't be able to continue rolling. It's not like it has like an infinitely long protein that wraps around and around and around while it's stuck. So something has to be going on here that's sort of interesting as far as affinity as well. The sign in this location is one of our proteins that's going to be involved in this interaction. So the quote-unquote sign is some molecule or protein that is going to mark that location, the ligand for which will be on our white blood cell. And so we'll have a receptor-ligand interaction that starts to slow that cell down as well as pulling it out of the center of that blood vessel. For this step of rolling / sticking, whatever we're going to call it, the interaction is between two things that are shown here, one of which is called a selectin. So a selectin is a specific type of protein, and the type of protein is called a lectin. Selectin. So, what is a lectin? This is the second time we've encountered a lectin this semester, so I told you what it was before. Yeah, Molly.
The thing that binds carbohydrates.
So lectin is a protein that binds carbohydrates. And selectins are a type of lectin. Therefore, what do you think a selectin ligand is? Some kind of carbohydrate, exactly. So our selectins are going to bind to some type of carbohydrate. And you can see this figure from your textbook where we have our selectin. It includes a lectin domain, and it's going to bind to some type of carbohydrate. Yeah, Jordan.
Is the selectin on the cell or on the vessel?
Great question. Let's come back to that in just 1 second. So this is going to be the first interaction that we see. In total, I usually think about 3 selectins. Your book puts a 4th one on there. I have always taught this in the past as there are 3 selectins. Your book does put up a 4th one. They are known as L-selectin, P-selectin, and E-selectin. L-selectin is the one that I know the most about, both because of using it in past research and it's the one that people talk about the most frequently. Its CD number is CD62L. For P-selectin, the CD number is CD62P. And for E-selectin, it's CD62E. And for reasons that have more to do with, psychology and psychoanalyzing me than anything else, a lot of times when I would hear research presentations, I would hear people talk about CD62 ligand, and then you're talking about CD62L, and the L here does not stand for ligand, and I would get really annoyed. Because in fact, the L in L-selectin, which is the same L that's in CD62L, stands for leukocyte. And in the case of an L-selectin mediated interaction, Jordan, this is on the leukocyte. L-selectin is on the leukocyte. P-selectin, the P in P-selectin stands for platelet. The E in E-selectin stands for endothelium. Typically, for the versions of this that I'm going to show you, P and E-selectin are kind of going to be the same. I'm going to have them be relatively interchangeable. And either of them are going to be on the epithelial cell wall or the site of inflammation. So the answer to your question in big picture terms, Jordan, is it selected on the cell or is it on the location? It depends on which selectin we're talking about. So with L-selectin, it's on the moving cell. With the others, it's on the location, the epithelial cell, this inflamed tissue. And each selectin will have some type of carbohydrate that it will bind. PSGL1 is one that's relatively famous. CD34 is another one that's famous. Really, you need to know that there's a carbohydrate ligand. And different selectins can have somewhat different ligands. So there's some kind of carbohydrate ligand There are many of them, and we can see nice interactions between our selectins and those carbohydrate ligands. And this rolling process looks something like this. Here I am showing you the rolling of a naive cell, like a naive T cell, along a blood vessel in a lymph node to get that cell to roll at the lymph node. And so in that case, no, actually I'm showing you a neutrophil because I can read slides. Showing you a neutrophil on a site of inflammation. You can see that the selectin is on the walls of the endothelium here. And you can see that there is this carbohydrate ligand, in this case it's one known as SLEX on neutrophil. This will bind, but this binding interaction will not be particularly strong. And so when this cell is contacted by the blood or something else, something that might have caused a force to shear it, it will in fact pull that cell off. The interaction will come off, but the cell will get caught again on another selectin. And that cell will basically slow down its velocity, so it's moving much slower, as well as that cell will be moved to the side because it's making these interactions with the selectin instead of being in the middle of the vessel, in the middle of the blood flow. I almost said that the cell would slow its roll, and then I thought that was going to be a really funny way of phrasing that. And so this is the first step of this process. This process looks something like this. I love this video. I'm not going to be able to show you all of it today because some of it is not relevant, but parts of it are the best. I'm actually going to show you the first 40 seconds.
Which attracts more immune cells to the area,
what?
attracts more leukocytes,
Stop.
and also activates the...
Let's get rid of this guy. Don't know who he is. And let's start with this.
While red blood cells are carried away at high velocity by a strong blood flow, leukocytes roll slowly on endothelial cells. P-selectins on endothelial cells interact with PSGL1, a glycoprotein on leukocyte microvilli. Leukocytes, pushed by the blood flow, adhere and roll on endothelial cells because existing interactions are broken, while new ones are formed. These interactions are possible because the extended extracellular domains of both proteins emerge from the extracellular matrix, which covers the surface of both cell types.
And then cell biology happens. So that's really what rolling looks like. Yep, Nadia.
Is that accurate to what selectins actually look like?
Yes. Everything in it. So this is an 8-minute video that has a lot of different cell biological processes, and all of it is drawn by scientific animators based on crystal structure, and so everything in it is accurate. Yeah. No, so the cell bio stuff isn't super relevant, but it actually gets the end of the video does the other steps of this process. So we'll zoom to the end of it later. But if you go to get in the middle and look at, say, the cytoskeleton parts or the Golgi parts, they're all accurate and they're so cool.
So are leukocytes constantly rolling and going at a slow speed, or is there some sort of on-off state for which they can selectively slow down at certain parts?
Great question. So let's think about the answer to that, because the answer to that is a little bit tricky. And I have it sort of set up at the end of the slides, but let's address part of it now conceptually. So let's first imagine a neutrophil. The neutrophil has will have a carbohydrate. This tissue has a selectin. And so Emilio's question is the carbohydrate always there? Is the selectin always there? Or are they induced at some period of time? That's really what this question is asking. So let's think about it from a perspective of how you would want it to work. So if you are a neutrophil do you want to be able to find sites of inflammation all the time? Or do you want to only be able, or do you want to have a signal to tell you to find a site of inflammation? Well, but do you want to have to wait? Are neutrophils fast or slow in their reactions? Fast. So if you're a neutrophil, do you want to be able to hear the call all the time? Yes. You're going to be kind of, as a neutrophil, you're sort of going to be listening out. Do any tissues tell me they're inflamed? And so in this situation, the carbohydrate is present on neutrophils all the time. However, if you are the tissue, do you want to be sending out the call to say, neutrophil, come here, all the time? What do you think, Malik?
No.
No?
Because you want it to be like selective, like if you actually have people.
Yeah. So in fact, the selectin is induced when this site has inflammation. Part of that inflammatory response with the PRRs will result in the production of this selectin by the inflammatory response. The neutrophils always have the carbohydrate so that they could bind to any site that puts out a call for neutrophils. And so here the selectin is inducible, the carbohydrate is not induced. When we have naive cells going to blood vessels, the naive cells have L-selectin on their surface. They have that on their surface when they are naive because they want to go to lymph nodes. So the lymph node always has a specific carbohydrate. The cells have the selectin when they're naive, and when they are no longer naive, they turn the selectin off. So typically, the selectin part of this is regulated, and the carbohydrate is less regulated, and is more frequently constitutive. So usually, whichever partner in this has the selectin is going to turn that selectin on and off when it needs a selectin on and off. The carbohydrate is generally going to be there all the time. One thing that's really good about addressing that question right now is it does make you think about the fact that in different situations, and this is also sort of coming back to Jordan's question, in different situations, the partners could be on the moving cell or the location. And we're going to see that a lot throughout this process, especially as we get to the next step. So our next step is listed here as activation. There are a few ways we can think about this step. And this step is going to seem sort of like a step I'm making you memorize and not super useful until we get to the third step. And when we get to the third step, you're going to be like, oh, now I get why there had to be a step before. But right at first, it can seem kind of... So this activation step is really a step that involves signaling through a type of molecule known as a chemokine and its paired receptor, the chemokine receptor. All chemokine receptors have 7 transmembrane domains. This is the closest image I've been able to get out of a textbook, and it has 6, and it really annoys me. So there should always be 7 transmembrane domains on our chemokine. And that chemokine receptor is what is known as a G-coupled protein receptor, which you guys learned about with Dr. Dunaway. The G-coupled protein receptor has the G protein subunits in the cytoplasm, G-alpha, G-beta, and G-gamma, which are shown here, that can lead to signaling along with GTP. So it's that same process that you learned about with Dr. Dunaway. And these chemokines in general can lead to changes in cells, like changes in actin polymerization or adhesive ability to stick to things, or other cytoskeletal rearrangements, all of which in general can lead to this process of chemotaxis. What is chemotaxis? Yeah, no. Movement. Specifically, it's movement in response to a stimulus. So movement towards or away from a stimulus. In fact, the word chemokine is really a shorter way of saying chemotactic cytokine. So chemokines can be thought of as a subset of cytokines, though people usually think of them as their own thing. but they're proteins that are always involved in cell movement, and that's because signaling through that chemokine receptor leads to all of these cytoskeletal changes and adhesive changes that can influence chemotaxis. But you can see that there are a number of different signals that can come from this chemokine. As with the selectins, there are many different chemokines. There's a lot more than the selectins. Selectins are only 3. There's a lot of chemokines. And there are a lot of chemokine receptors as well. The chemokines can be divided into a few different classes based on their structures and based on where cysteines can be found in their structures and how those cysteines are grouped. So you can see that some of them have a structure as is shown at the top, and they might have a name that's something like CXC. Some of them have a structure that's the second one and have a name called CC. Some of them have a thing like this and are called XC. Some of them are CX3C based on this structure. And the way that we name them is simply based on those groups and then with a number. One other thing that you will notice is all of the names have an L in them. So for example, chemokines with this kind of structure might be named things like CXCL1 or CXCL2 or CXCL3. Those with this kind of structure might be named things like CCL1 or CCL2 or CCL3 all the way up to 25. There are also a couple XCL1, XCL2, and there's CXCCL1. The L in all of these cases stands for ligand. So if you see a name that looks like this, you know it's a chemokine. So the chemokine names usually stand out pretty well. And if there's an L, that means it's the chemokine. That means it's the ligand in the name. Each of these chemokines will bind to a specific receptor. Those chemokine receptors can be like those shown here, and their naming conventions look like this. They might be things like CCR1. The R in these cases stands for receptor, whereas the L stood for ligand. And there are receptors that correspond to many of the different structural classes that you saw for the chemokines. If life made sense, then the numbers would match. So CCR5 would bind to CCL5, but it doesn't totally work like that because obviously it can't be that simple. But there's always some pairing between a chemokine. As you will notice here, our leukocytes have chemokine receptors, and each type of leukocyte will have a different set of chemokine receptors. That may be based on, say, the identity of that cell as an eosinophil, or it may be due to the differentiation or activation state. So here you can see that a resting T cell and an activated T cell will have slightly different chemokine receptors. And this allows each of these types of cells to go to locations that are appropriate for that cell type. One other thing that we often talk about when we talk about this process is, I told you that there are kind of 3 steps. And you can also think about this process as being in some ways reminiscent of area codes. If a neutrophil wants to go to the skin, it needs to have the right carbohydrate to bind to the correct selectin, and then it needs to have a skin chemokine receptor. It's possible that a cell could start rolling and not have the right chemokine receptor for that location, and then be done, and not do the rest of the steps. So this is sort of allowing for a location specification system. So just like if you start dialing a phone number with a 9, you could mean 973, like here, or you could mean 919, like North Carolina. You don't know. It's not until you have the specification of the 7 versus the 1, and the 3 versus the 9. So we have this 3-step system that basically each part of each anatomic location has a unique type of selectin carbohydrate pair. There aren't a ton of them because there's only 3 kinds of selectins, but then a unique chemokine receptor pair that specifies that location and helps a cell go to that specific location as opposed to some other location. And just like we can distinguish the whole United States just with 3 numbers, we can distinguish all the anatomic locations with the proteins that are involved in the 3 steps. So it's easier to talk about this here because there's a lot of variants as opposed to with the selectins where there's only 3, and you're like, this doesn't make as much sense. So this will allow each of our different types of leukocytes to start to traffic to locations of interest. The next step is a step known as, that has a few different names. It's listed here as either arrest or adhesion. Sometimes you will see it listed as firm arrest. Sometimes you will see it listed as stopping. So I told you that originally I learned the first one as sticking, and then I learned this one as stopping. Either way, it doesn't matter. I probably will use those terms interchangeably along the slides because, that's how I work. But if you imagine this step, before we talk through this step, what do we think this cell is going to do in this step? Stop. So instead of having that cell rolling at a slow speed, the cell is going to come to an absolute, complete stop at this point. Remember that this is probably going to be quite dangerous for the cell. So you don't want the cell to be able to stop all the time. Imagine what would happen if cells just stopped by mistake in your blood vessels. You'd end up with torn up cells and clogs in your vessels pretty quickly. And so while rolling, it's not a huge deal if cells roll by mistake. It's a really big problem if cells stop by mistake. And so stopping has a little bit more complexity going on with it. If you think about this from a biochemistry point of view, what might you think about the proteins that are involved in this process compared to, say, the selected carbohydrate interaction? Yeah, Jordan.
Higher affinity.
Higher affinity. In fact, extremely high affinity. This affinity is such that it can pull that cell to a complete stop. The interacting molecules in question are known as integrins. And integrins typically bind to molecules that are known as cell adhesion molecules, or CAMs. Although if you looked closely at some of the selectin ligands, some of them also had CAM in their name. So the names of some of these ligands are a little tricky. You can sort of think of these as being integrin ligands in some cases. And so you can see that we have some kind of integrin ligand, like ICAM1, which is particularly famous, this binding to an integrin. Notice that our integrin, in both versions of this figure, has 2 chains, has 2 parts. So all integrins have an alpha chain and a beta chain. And the specific integrin and the specific binding of that integrin is based on the combination of the alpha chain and the beta chain. You can see, for example, that the names of many of our integrins have alpha something, beta something, where we're giving a number or a letter that tells us which alpha chain and which beta chain. Many of them also have a name that they're commonly known as. So alpha M beta 2, I think that may be the first time in my entire life that I said alpha M beta 2. I always just say MAC1. I'm not sure that I've ever said alpha L beta 2 either. I usually say LFA1. But I have I always call alpha 4 beta 7 alpha 4 beta 7. I actually had never heard of it as LPAM1 until looking at this image. So different ones can have sort of different interchangeable names. There are also in many cases CD names for these, but this is one of the places where CD nomenclature hasn't sort of taken over in people's minds, and people still tend to use these types of names. The integrins are binding to Ig superfamily members. Most famously, there are things like ICAM 1, 2, or 3, VCAM 1, LFA 1, 2, or MADCAM. MADCAM is for mucosal. Is the mucosal addressant. VCAM is vascular. So they can be found in a number of different places throughout this process. So there's the other really critical thing to know about integrins has to do with that high affinity problem, or not really high affinity, but the fact that they have crazy high affinity, but also that they're very dangerous. So on all of your T cells right now, you don't necessarily want them having integrins all over the place to potentially stick places incorrectly. And the way that this is worked out is that integrins actually have two types of folding. They have two conformations that they can undertake. One of them is a bent conformation, where you can see that the integrin molecule has its transmembrane domain and then bends and sort of goes back towards the membrane. Here you can see that in ribbon diagram, where the integrin is actually bent back upon itself. Sometimes this is referred to as the jackknife conformation, where this integrin is, wrapped inside like a Swiss Army knife that you pull out. And then in other times, the integrin has its extended conformation, where the entire protein is extended in this way, and you can see that from our textbook on the right-hand side. Only the protein with the extended conformation is able to bind to ICAM with any kind of appreciable affinity, because that binding site is here at the tip of the alpha and beta domains. That's where we bind to ICAM. You can see that that binding site is sort of hidden when the integrin is in the jackknife conformation, and only when that integrin is actually extended is that integrin able to bind to its ligand with high affinity. And so you might wonder, well, what allows our integrin to open and to become able to bind to ICAM? And the answer is that's one of the effects of the chemokine signaling. When the cell gets signaling through the chemokine receptor, that tells the cell to release the integrins. What you might also realize from this is that the cell with the chemokine receptor has to be the same cell that has the integrin. So just like we can think about, well, which one makes the chemokine, which one has the chemokine receptor, which one has the selectin, which one has the carbohydrate, which one has the integrin, which one has the integrin ligand. It can go in all sorts of different patterns, but the one with the chemokine receptor better be the one with the integrin, because you gotta get a signal from the chemokine receptor to turn the integrin on. Doesn't work otherwise. The integrin would just be down forever if there was no chemokine receptor and no chemokine signal. There has to be a chemokine signal to activate that integrin. Of course, you don't have to just believe me on this. All right, this is close enough.
The vesicle fuses with the plasma membrane. Proteins contained in the vesicle's lumen are secreted, and proteins embedded in the vesicle's membrane diffuse in the cell membrane. At sites of inflammation, chemokines secreted by endothelial cells bind to the extracellular domains of G-protein-coupled membrane receptors. This binding causes a conformation change in the cytosolic portion of the receptor, and the consequent activation of a subunit of the G protein. The activation of the G protein subunit triggers a cascade of protein activation, which in turn leads to the activation and clustering of integrins inside lipid rafts. A dramatic conformational change occurs in the extracellular domain of the activated integrins. This now allows for their interaction with ICAM proteins displayed at the surface of the endothelial cells. These strong interactions immobilize the rolling leukocyte at the site of inflammation. Additional signaling events cause a profound reorganization of the cytoskeleton, resulting in the spreading of one edge of the leukocyte. The leading edge of the leukocyte inserts itself between endothelial cells, and the leukocyte migrates through the blood vessel wall into the inflamed tissue. Rolling, activation, adhesion, and transendothelial migration are the 4 steps of a process called leukocyte extravasation.
Thank you, narrator man. He does, except that for some of the cell bio parts in the middle that I skipped over, he pronounces the names of the organelles wrong. And I get really annoyed. Huh? No, I can't remember what it is now, but there's something where I'm just like, no, that's not how you pronounce that word. So, in any case, that's really what this whole process looks like. So the final step is that we're going to have our cell actually pulled to a stop by that integrin cell adhesion molecule interaction. That integrin was activated by activation with our chemokine receptor signaling. And before our cell can actually get any of those signals, it had to be slowed down through this rolling process. At all 3 steps, there are multiple different potential proteins that can be acting, and so this is where we get that area code-like thing, where the specific selectin and carbohydrate, the specific chemokine receptor, the specific integrin ligand dictate which part of the body, which anatomic location we're at, and help the cell know where it is and where it should be moving to. The final part of this process you saw in that video, and that final part of the process is where the cell actually moves in between epithelial cell layers to leave the vessel and go to the surrounding tissue. One thing that you saw from the video was this required a massive cytoskeletal rearrangement in that cell to flatten it so that it can squeeze between epithelial cell walls. We know considerably less about this process and how this process works. So this process is known as listed here, diapedesis. It is sometimes also listed as transendothelial migration or extravasation, leaving the vasculature, extravasation, or transendothelial migration, moving across the endothelial cell layer. What you should notice here is our leukocyte moving across the endothelial cell, and that leukocyte has to dramatically reshape itself. So here is our leukocyte, and you can see how it's completely different shaped, and that's the nicest way I can phrase it, to squeeze in between that endothelial cell layer. As I mentioned before, there is considerably less known about this part of the process than is known about the other parts of the process. And so all I can really tell you for this one is that there seem to be a number of chemokines and chemokine receptors involved in this process as well. But other parts of it are not well understood. Yes, Molly.
I have a question, and the answer is probably because it's in 3 dimensions and not a picture.
Okay.
But the basement membrane of endothelial tissue is contiguous.
Mhm.
So, like, in order to escape, does it have to deal with the basement membrane, or can it just go like out because it's 3D and not flat?
So that's a great question. And the answer to that question is really it depends. there are some places where that membrane is specialized to allow cells through. There are other places where that cell might need to have something like a protease to help it cut through the layer. That process is a process that some cells have to do to cut through different layers in order to move. Those proteases, the most famous of which are called matrix metalloproteases, they cut the extracellular matrix, so they're matrix proteases. Those matrix metalloproteases are actually a huge deal in cancer biology because tumors really have to be able to cut membranes to allow themselves to metastasize. And so those cells are used for some normal processes. This could be one of the normal processes where cells use them, but those proteases can also be pretty dramatically, have their regulation changed in some pathologic states. But inherent in my answer to Molly's question was the fact that all trafficking is not equal. Trafficking to each of these sites is similar in that it has the same 3 steps, but the individual participants in those steps vary based on the location that our cell is going to. And so now I want to go through this process, not in general terms, but in the specific terms of different scenarios. So the big scenarios where this can happen are scenarios where our neutrophil or our monocyte can migrate to a site of infection or tissue injury, that initial inflammation step. You can see our monocyte or neutrophil leaving the vessel wall here. Or we can imagine our naive B cell or T cell wanting to leave the blood vessel to enter a lymph node so that they can first encounter antigen. And really the reason why we're talking about this whole process at this point of the semester is because we are going to be dealing with our naive T cell trying to encounter antigen, and so this is kind of the one, that's why we're talking about all this now. One could also imagine that after our T cell sees antigen in the lymph node, it doesn't really matter if it can combat the antigen in the lymph node. It has to combat the antigen wherever the real infection is. You don't just get infections of your lymph node. When you have a rash, that's an infection of your skin, etc. And so the T cell will then have to go to that location and will have to, again, leave the vessel to deal with the site of infection. I am largely going to talk about these 2 processes on the left. I'll say a couple things about some details about this one on the right, but I'm not going to go through that one in great detail, and that's because how a T cell goes to the skin, and how a T cell goes to the liver, and how a T cell goes to the lung, and how a T cell goes to whatever else, it's all going to be a little bit different. So I can't generalize it in quite the same way. So your textbook also describes these pretty well. And so for any of the situations that we're talking about, we can think about what are the selectin carbohydrate pair members. We can think about what are the chemokine and chemokine receptor members. And we can think about what are the integrin ligand members. And first we can imagine a neutrophil going to inflamed tissue. And suddenly I don't know how to spell inflamed. So we can imagine first this situation. For the first step of this process, you can see your book lists PSGL1. It actually lists 2 things, but I covered one of them up with a white box because I only wanted you to think about PSGL1. PSGL1 is the carbohydrate. and it is found on the neutrophil all the time. The ligand of PSGL1 can be either P or E selected. And this is on the inflamed tissue. It is induced by inflammation. So, nice happy tissue does not have a P-selectin or E-selectin on its surface. But when tissue starts to have an inflammatory response, those endothelial cells will start expressing P and E-selectin to recruit in neutrophils, which have the carbohydrate ligand PSGL1. So this is for this specific situation. Then we get to the chemokine receptor part of this. This one, there's one thing that always is slightly annoying here. The chemokine in this case is also made by the inflamed tissue. So most tissue does not make this chemokine. But when tissue gets inflamed, it starts making this chemokine to say, hello, neutrophils, come here. And so again, this is going to be an inducible event to call in the neutrophils. And the chemokine in this case has the frustrating name of IL-8. It's the only chemokine with an IL name, because it was one of the first ones described and nobody knew what chemokine were. So they just called it IL-8. So IL-8 is the chemokine. And again, it's made on the inflamed tissue in response to inflammation, just like the P and E-selectin was made by the inflamed tissue in response to inflammation. Because the naming is just awesome, you can imagine this being the IL-8 receptor as our chemokine receptor. This is on the neutrophil, and it's also on the neutrophil basically all the time, because the neutrophil really wants to be able to go to this inflamed site ASAP. You don't want the neutrophil to have to turn stuff on. You want the neutrophil to be able to respond really, really quickly. Given that this neutrophil has our chemokine receptor, you can figure out that our integrin must be on the neutrophil, because that allows for the signaling to happen. Typically, the integrin ligand here is ICAM, or the integrin ligand is ICAM1, and the integrin here is LFA1, to allow this trafficking process to happen. So our ligand is ICAM1, it is on the inflamed tissue, and LFA1 is our integrin, it's on the neutrophil. It only is in its high affinity form after receiving signals through the IL-8 receptor. And so this is what this process looks like for a neutrophil going to a site of inflammation. You can see this same process here from an older version of your textbook. So we have our neutrophil. Our neutrophil is going to have some carbohydrate ligands as well as a chemokine receptor on its surface all the time. When this area gets inflammation, perhaps that's because of cytokines, perhaps that's because of PRR signals, this area will turn on its P and E selectins, its CAMs, as well as its chemokine production, its IL-8 production. So we'll start getting our rolling through this selection interaction. IL-8 will signal the IL-8 receptor that will turn on our integrins. Why they show these two different versions I don't really like. And then finally our active integrin will bind to the CAM. Our cell will be stopped in its tracks and will be ready to go through this cell wall. So this is how this process looks for a site of inflammation. So we just saw this site of inflammation or process, but we can also think about what happens when our naive cell wants to traffic to find antigen. T cells like to traffic to different locations based on their differentiation state. A naive T cell, if you were a naive T cell, what would your goal in life be as a naive T cell? What would your purpose be? I ask you about your purpose in lab every week. What would your purpose be as a naive T cell? Yeah,
To get to a secondary lymphoid organ so you can start like seeing stuff and being specified.
okay.
Oh,
So-
they're already specified.
Yes, so they've already done BDJ, so they already have a specific receptor, but what do they want to do? Like, really simple. What is their goal? Huh? They want to bind what? So they want to bind their antigen. They want to bind their matching antigen. So really what they want to do is they want to look around the body and find their antigen. You could imagine one thing they could do is they could start here in your head, and they could look at this cell and be like, hello, do you have my antigen? And they go, hello, do you have my antigen? Hello, do you have my antigen? And they could go through every single cell of your whole body, one after the other. That would be rather inefficient. However, since we have this nice system of lymph nodes and other secondary lymphoid organs collecting things from different anatomic locations, the alternative is that they can just make a circuit of the lymph nodes and look and say, does your area have my antigen? Does your area have my antigen? Does your area have my antigen? So those naive T cells really want to simply enter lymph nodes to look for their antigen. They want to make a circuit of the lymph nodes. And so for a naive T cell, it's really important to go to a lymph node. And so the red cells here are the naive T cells. And so you can see the naive T cells can travel through the blood, and then they're going to go to the lymph node. They might come back to the blood, or they might just go lymph node to lymph node to lymph node to lymph node. So, but here, our T cell finds its antigen. And it becomes activated. It becomes blue. Now, what is that cell's purpose? Yes, Amelia.
Scour the anatomical area to find its antigen.
So now it wants to leave the lymph node and find its antigen. Happily, it knows which region of the body to look in. So look in the region of the body that corresponds to that lymph node. What that means is the first part of this whole idea of how a T cell response should work partially involves differences in how trafficking happens. How do you think the cell is able to do this process of, what do you think happens in terms of trafficking molecules between the red cell and the blue cell, between these two parts of the T cell's life? What generally is going to happen with the trafficking molecules? Yeah, Molly?
Don't they have to change so that they can go to different locations?
Yeah, they have to delta. They have to change. So there's going to be some kind of change. So we're at the beginning, naive T cells probably have the lymph node kind of trafficking molecules. And as part of this activation process, the cell is going to switch its trafficking molecules so that it now doesn't have the lymph node ones anymore. So it doesn't keep going to the lymph node, and instead has some that are specific to its location where it wants to go to. When we think about the structure of the lymph nodes have one particularly important structural feature that we care about here. In our lymph nodes, I've told you before that there are vessels, blood vessels, coming in and out. You can see such a vessel here. But in fact, there is one specific type of vessel that really matters here. That type of vessel is called a high endothelial venule, or an HEV. You can imagine this is a very small vein, because it's a venule. But it also is somehow special because it has something because of high endothelial. And this one I actually didn't understand for a long time. So I like this picture from your textbook because it kind of makes the part of high endothelial clear. High endothelial venules are so called because they have endothelial cells that are really tall. They're tall and skinny, so they're high. They go up high. And so you can see these cells are sort of rectangular in this way. They're skinny and kind of tall. And so these are high endothelial venules. They're specialized. They're venules that have this special set of endothelial cells that are really tall. And so the HEV is basically where our naive T cell is going to enter into the lymph node. And so the HEV is sort of the tissue in this case. And so instead of thinking about the neutrophil and the inflamed tissue, we can think about a naive lymphocyte going to an HEV. Again, there are specific members of this partnership that are expressed on each of these proteins. The lymphocyte in this case has the selectin. The lymphocyte has L-selectin. The selectin is found only on naive cells. It gets turned off with activation. So the selectin, again, just like in the past scenario, is the one that's going on and off. It's just going on and off, not quite as narrow of a time. The whole time this cell is naive, it has L-selectin. Later on, it gets rid of it when it gets activated. Yep, no, okay.
Figure it out.
The HEV has the specific carbohydrate on here. The carbohydrate in this case is one called CD34. Do you think that HEVs turn CD34 on and off, or do you think that HEVs have CD34 all the time? What do you think, Marina? All the time. Why all the time? Yeah, lymph nodes always want T cells to come. Lymph nodes always want T cells to be finding them. And so CD34 is going to be on permanently. L-selectin is going to be turned on when the cell is naive and turned off when the cell is then activated. Now we're going to have the chemokine co-receptor interaction. The chemokine receptor in this case is CCR7, and it is on the naive cell. The CCR7 ligands are CCL19 and CCL21. I was checking to see what your textbook called them before I gave you the wrong name. So typically we see CCL19 and CCL21 binding to CCR7. These do have other names that they are commonly known as, but CCL19, CCL21 sound great for us. And they bind to CCR7. CCR7, again, is always on that cell when it's naive. It gets turned off when the cell gets activated. I have done a whole lot of CD62L, CCR7 flow cytometry on cells help me learn, are they naive cells or are they activated cells? Because if the cell is a naive cell, it should have these two things. If the cell is an activated cell, it should be missing these two things. The HEV, again, is making CCL19 and CCL21 all the time, because it always wants to call in naive lymphocytes. Then we have our integrin ligands. You can see those integrin ligands listed here. Specifically, you can see that ICAM, our integrin ligand, is on the HEV, and you can see that LFA1, our integrin, is on our T cell. Our integrin, of course, has to be on the cell with the chemokine receptor so that we can get signaling through the chemokine receptor to turn on that cell. Once each of these interactions happen, our cell will be stopped, and then we'll move through the barrier between our two cell types. And so this is what that naive cell trafficking to the lymph node looks like. When that cell becomes activated, the biggest things that happen to it are that it loses L-selectin and it loses CCR7. Then if we think about that lymph node like our lymphoid organ, we can go back to the fact that I told you that there are different areas within that secondary lymphoid organ. So there are T cell zones and B cell zones and all sorts of different zones there. So I've told you how our cell, in the case of our naive T cell, gets to the secondary lymphoid organ, but I haven't told you how does it know where to go once it's in the secondary lymphoid organ. The answer to that question is that stromal cells, structural cells in different parts of the secondary lymphoid organ make different chemokines. And those chemokines call in the appropriate cells. So you can see that, for example, here, CCL19, or actually, here CCL19 and CCL21 are being made. Here, CXL13 is being made. That calls in B cells. You can see the T cells are being called in by a different chemokine. And it's the same chemokines in the lymph node that it is down here displayed. So there are going to be unique chemokines made throughout that secondary lymphoid organ that are going to help the cells sort themselves out once they get into the secondary lymphoid organ. So that's sort of the general part of this process. There are people with interesting mutations in many of these different things where they basically have T cells, but they can't fight infections because their T cells can't ever traffic to the right location. The most famous of them were integrin deficient people, but then there was later found to be a group of people who had problems adding carbohydrates to the surface of their cells. And those people not only cannot do blood cell trafficking, they also have no blood type because your blood type is also based on the carbohydrates on the surface of the cells. It's a glycosyl-transferase problem. I think there are like 5 people in the world who have ever been described to have this. And if I remember correctly, also cows. Mutations in these processes can be really difficult because, again, it looks like you're fine, it looks like you have plenty of T cells, but your T cells are never able to actually do anything. And so it's been an interesting set of diseases to study. And we'll start talking about activation on Monday. Monday is really going to be signal transduction time. So be aware, be ready for that. I will see you then. Have a great weekend.